Eukaryotic cells and microorganisms have a number of medically important organisms in them. Eukaryotic cells are all in the domain Eukarya. Their distinguishing feature is they have a membrane-bound nucleus and numerous membranous organelles. Many eukaryotic cells lack cell walls, such as animals and most protozoans. Some may have a glycocalyx. Just like the prokaryotic cell, this is a sticky mass of macromolecules. But unlike the prokaryotic cell glycocalyx, this one is covalently bound to the cytoplasmic membrane. The glycocalyx helps anchor animal cells to each other. It strengthens the cell surface, and it does provide some protection from drying. It's important in cell-to-cell recognition and communication in eukaryotic cells. Cells that have a cell wall do not have a glycocalyx. Fungi, algae, and plants have cell walls. Cell walls provide protection from the environment, provide shape for the organism, and protect against osmotic pressure changes. Cell walls are composed of various polysaccharides. In plants, the major polysaccharide in the cell wall is cellulose. Fungi will have cellulose, chitin, and/or glucomannan in their cell walls. And algae have a host of different chemicals in their cell walls. They may have cellulose, various proteins, agar, carrageenan, silicates, algin, and calcium carbonate. All eukaryotic cells have a cell membrane, and it's the basic phospholipid bilayer that is the fluid mosaic model that we saw in prokaryotic cells. However, there are many more proteins in the eukaryotic cell wall. These proteins act as recognition molecules, enzymes, receptors, and carriers or channels. There are many more carrier channels for facilitated diffusion in eukaryotic cells than in prokaryotic cells. Also, multicellular organisms that are eukaryotic have proteins that anchor cells to each other. Eukaryotic cell membranes contain sterols to strengthen and maintain membrane fluidity. Also, in eukaryotic cells, we see membrane rafts. These are small areas of lipids and proteins that seem to stay together. These are used to localize cellular processes, things like signaling inside the cell or protein sorting, and sometimes they're involved in some types of cell movement. However, because these membrane rafts are a stable collection of molecules, they can be used as entryways for some viruses. Also, eukaryotic cell membranes have a number of exterior polysaccharide molecules that are used for cell signaling. The cell membrane function still controls the movement of materials into and out of the cell, and the eukaryotic cell uses the same passive processes as the prokaryotic cell: diffusion, facilitated diffusion, and osmosis. Also, in terms of active processes, pumps are still a method for moving molecules across the membrane, but eukaryotic cells use endocytosis. This is a way of taking larger molecules into the cell. If the molecule is dissolved in a liquid, we refer to the endocytosis as pinocytosis. If it's a solid particle, we call it phagocytosis. The reverse of endocytosis is exocytosis. This works because the cell membrane is fluid. That is, when the membrane touches itself, it just kind of coalesces into a separate membrane. So here, we see molecules coming in. The membrane is sort of invaginating here, and molecules are coming into this area. When the membrane touches right here, we're going to pinch off this vesicle, and a vesicle is simply a membrane-bound sac that holds something. The membrane here will have remained solid. It will not have broken at any point. Exocytosis is the reverse of this. A vesicle inside the cell will move up to the cell membrane, touch it, and become a part of the outside of the cell membrane, and whatever was inside the vesicle will be released to the outside. Here you see an amoeba engulfing a paramecium. The amoeba sends out pseudopods surrounding the paramecium, and here where it's touching, the membrane will coalesce, and we will have the exterior membrane stay solid, and we will form this interior vesicle around the paramecium. Eukaryotic cytoplasm is chemically more complex. It contains both non-membranous and membranous organelles. Eukaryotic cells have organelles for motility. The flagella in eukaryotic cells have a whip-like movement rather than that rotary movement. Also, the flagella is an interior part of the cell. It is surrounded by the cell membrane. The shaft is much more complicated. It's composed of multiple tubules that are made of the protein tubulin. Basically, the individual tubes are arranged in 9 pair in a circle around a central 2 pair of tubules. Flagella act the same way, allowing for positive and negative phototaxis and chemotaxis in eukaryotic cells. Now here you see the flagella, a cross section. Here's the membrane that surrounds it. That's different than in the prokaryotic cell, which did not have a membrane around it. And here you see the individual tubules. There are 9 pair arranged in a circle around a central pair. These tubules can slide up and down on each other, and this is what allows for the whip-like movement of the eukaryotic flagella. Cilia are shorter and more numerous structures that tend to cover the surface of the cell. They still have the 9+2 arrangement of tubules, and they're still made out of tubulin. They are also involved in cellular motility, and they can move material across a cell surface. Some of the other non-membranous organelles are the ribosomes. The ribosomes are still involved in protein synthesis, but the ribosomes of eukaryotic cells are a little different than the ribosomes of prokaryotic cells. A man named Svedberg came up with a sedimentation technique, and he labeled each layer a Svedberg unit. The ribosomes of eukaryotic cells are larger. They're at the 80S level. The prokaryotic ribosomes come out at the 70S level. So while they have the same function, that of protein synthesis, they are structurally a little different. Ribosomes in the eukaryotic cell may be attached to the endoplasmic reticulum, and they're called bound ribosomes, or they may be free in the cytoplasm. The cytoskeleton is an internal scaffolding of fibers and tubules. These help anchor the organelles. Also, they're involved in cytoplasmic streaming. That is, they help direct the cytoplasm around the cell. They help the cytoplasmic membrane move. This is one of the ways cells can make pseudopods. They also provide cell shape to those cells that do not have a cell wall. Centrioles are found only in animal cells and some fungal cells. The region of the cytoplasm where they're found is called the centrosome. Centrioles play a role in mitosis, cytokinesis, and the formation of cilia and flagella. But because not all eukaryotic cells have centrioles, and yet they do undergo mitosis, cytokinesis, and have cilia and flagella, these organelles are still under continuing research. The membranous organelles will start with the nucleus. The nucleus is the control center of the cell. The fluid inside the nucleus is known as the nucleoplasm, and the nucleus contains the chromatin, which is arranged in chromosomes. Chromosomes are DNA and the associated proteins that help arrange the DNA. Also in the nucleus are one or more structures. A singular is a nucleolus, multiple are nucleoli. The nucleoli are where RNA synthesis occurs. This is where the ribosomes are made. The nucleus is surrounded by a nuclear envelope, and this is a double membrane, and there are large holes in this membrane called nuclear pores. Things do not freely move from the cytoplasm to the nucleoplasm. For things to get through the nuclear pores, they have to have a special tag. They have to be recognized as something that can either enter or leave the nucleus. The endoplasmic reticulum is a transport system. It is a membranous series of tubes and channels that's continuous with the nuclear membrane. The endoplasmic reticulum acts as a site for enzyme attachment. It also helps divide the cytoplasm up into separate room-like areas so that chemical reactions that are going on in the cytoplasm don't interfere with each other. Smooth endoplasmic reticulum does not have ribosomes attached to it. This is the site of lipid synthesis and transport. Rough endoplasmic reticulum has ribosomes attached to it. Here we make complex proteins that may be exported from the cell. The Golgi body is the exporting part of the cell. It will receive, process, and package large molecules for release from the cell. It prepares secretory vesicles, little sacs that contain these molecules that will travel to the cell membrane, and through the process of exocytosis, the material will be released from the cell. Lysosomes are sacs of catabolic enzymes. This is basically the cell's digestive system. Also, lysosomes are sometimes called suicide sacs. They will self-destruct old, damaged, or diseased cells. Vacuole is simply a general term for sac. Usually, vacuoles inside cells are storing some sort of material, water, glycogen, or something the cell needs for later. Here you see the inside of a cell, the smooth endoplasmic reticulum, and as it manufactures things, it will pinch off little transport vesicles that will go to the Golgi body here. Now remember, all of these membranes are just like the cell membrane. They're very fluid. So when the membrane touches, it either pinches off or fuses to another membrane. As molecules pass through the Golgi apparatus, they are processed and modified, and these secretory vesicles are released and travel to the surface of the cell, where the material inside the vesicle is released to the outside. Here we see endocytosis, and since we're taking in a particle, it's technically phagocytosis. This is then the food vesicle or food vacuole that contains the material that was ingested. A lysosome, and notice that the lysosomes come off the Golgi body, will then fuse with the food vacuole. The digestive enzymes will break down whatever material is in the vacuole. This will be used for energy, and any leftovers will go ahead and be transported to the cell surface so that the material that was not used can be excreted from the cell. The mitochondria is another membranous organelle. Now this particular organelle has a double membrane, an inner and an outer membrane. The mitochondria is where ATP is produced, so the mitochondria is sometimes referred to as the powerhouse of the cell. The mitochondria are interesting organelles because they contain 70S ribosomes and circular molecules of DNA, just like prokaryotic cells. Also, the mitochondria can divide independent of cell division. All other organelles in the cell divide only prior to the cell dividing. Mitochondria can divide in response to cellular demand for ATP. Chloroplasts are the site of photosynthesis. All cells will have mitochondria, but only photosynthetic cells will have chloroplasts. They too have a double membrane, an outer and an inner membrane, and they also contain 70S ribosomes and circular molecular DNA, just like prokaryotic cells. They too can divide independent of cell division. This leads us to the endosymbiotic theory. Because mitochondria and chloroplasts are semi-autonomous, that is, they do their own cell division when they need to, and both of them contain ribosomes and DNA that is similar to prokaryotic cells, some scientists feel that eukaryotic cells were formed from the union of small aerobic prokaryotes with larger anaerobic prokaryotes. The smaller prokaryotes were ingested by the larger cell, but they weren't digested. Rather, they stayed in that vesicular intake membrane, so that the inner membrane of mitochondria and chloroplasts were originally the cell membrane of the prokaryotic cell, and the outer membrane is the vesicle that was formed when they were ingested by the larger cell. Over time, it's felt that the larger cell became dependent on this smaller parasite, and they began to coexist as one. As we look at the eukaryotic organisms of medical importance, we're going to be looking at the protozoa, the fungi, the helminths, and the arthropods. Now, as we start talking about eukaryotes, let's talk about reproduction. Eukaryotes have multiple chromosomes and they're linear. Anytime a cell divides, an exact copy of its DNA needs to be passed on to the offspring. So all of these linear chromosomes have to be copied before the new cell divides. And we have to make sure that we divide them up in such a way that each new cell gets one copy of the old material. This makes reproduction and cell division much more complicated in eukaryotic cells. and it's divided into two pieces, nuclear division and cytoplasmic division. Eukaryotic cells can reproduce asexually by binary fission, simply splitting in two, budding, pinching off a little part of the cell, fragmentation, spore formation, or a process called schizogony that we'll talk about in a minute. Eukaryotic cells may also reproduce sexually. Here the cells will make gametes, the two gametes will fuse, and a zygote will develop. And some eukaryotic organisms use both sexual and asexual reproduction. Algae, fungi, and some protozoa do this. The nuclear division is aimed at keeping the chromosome number constant for an organism. Now, an organism is said to be diploid if there are two copies of each chromosome. We sometimes represent this as 2n. An organism is said to be haploid if there's only one copy of each chromosome. We represent this as 1n. Some cells are normally haploid. Other cells are normally diploid. Prior to cell division, each chromosome is copied. Mitosis is the nuclear division that maintains the original number of chromosomes, so that if an organism was diploid, it remains diploid. If it was haploid, it remains haploid. This is a single nuclear division where you start with one cell and you get two new identical cells. Meiosis is a nuclear division that results in half the number of original chromosomes. Diploid cells will give rise to haploid cells. Now, meiosis is a two-step process that divides the chromosomes so that one diploid cell ends up yielding 4 haploid cells. Cytokinesis is the division of the cytoplasm. Now, this may occur simultaneously with the final stage of nuclear division. It may be postponed for a while and the cell may stay multinucleate, or it may not occur at all and the cell may remain multinucleate for the rest of its existence. Schizogony is a process that's seen in some protozoa. It's a form of asexual reproduction. The nucleus undergoes multiple mitoses. A large multinucleate cell is formed that's known as a schizont. Then at some time, cytokinesis occurs simultaneously and numerous uninucleate cells are released. Let's look first at the protozoa. Now the protozoa are in the kingdom Protista. And in Protista we have protozoa and algae. Because the algae are photosynthetic and make their own food, they are not important to us medically. They're important environmentally, but we won't be talking about them. We'll only talk about the protozoa in this kingdom. Protozoa require a moist environment. Most of them are free living. That is, they get all the nutrients they need from the environment, but a few are pathogens. They have two basic stages in their life cycle. There's the motile feeding stage, known as the trophozoite or the troph, and then there is the hardy resting stage, known as the cyst. When these organisms find themselves with plenty of food and a nice temperature, they exist in the trophozoite stage. If food or temperature become unfavorable, they will go into this more dormant resting stage called a cyst that will allow them to survive the more adverse conditions. Protozoa are heterotrophic. This means they live off of organic molecules that are produced by other organisms. Most protozoa reproduce asexually by binary fission or by schizogony. Some will exchange DNA in a process called conjugation. The new classification using rRNA gives you several classes, but we're going to use the older common classification that was based on motility. In this classification system, protozoa are placed into 4 classes depending on what kind of organelle they have for motility. Sarcodina move by pseudopods. Examples here are the amoeba, Naegleria, acanthamoeba, and entamoeba. Naegleria and acanthamoeba are free-living organisms that become accidental pathogens. People can get Naegleria from swimming in brackish water. Unfortunately, relatively young people get this. It goes to the brain and is almost always fatal. Acanthamoeba tends to settle more in the eye, where it causes diseases of the cornea, and Entamoeba is responsible for Montezuma's revenge. This gives you a fearful diarrhea if you ingest the cysts in contaminated water. Mastigophora move using flagella. Examples here are Trichomonas, Giardia, Trypanosoma, and Leishmania. Trichomonas lives in the vagina, where it can cause trichomoniasis, a rather unpleasant, uncomfortable disease. Giardia causes a lovely form of diarrhea. Giardia lives in natural water, and people can get it particularly when they go camping. Trypanosoma and Leishmania are blood and tissue parasites. These are typically acquired from being bitten by certain insects, and these are seen more in tropical climates. Ciliophora move with cilia. There is only one ciliate that is pathogenic for humans, and that's Balantidium coli. It causes diarrhea. It's associated with the ingestion of material contaminated with pig feces. And the sporozoa have no organelle for motility. These are things like Plasmodia, Toxoplasma, and Cryptosporidium. Plasmodium causes malaria, and it gets around based on a mosquito transferring it. Toxoplasma spores can be ingested if people come in contact with infected cat feces. Toxoplasma typically does not give anyone a severe disease unless they're pregnant or immunocompromised. Toxoplasma is why pregnant women are told not to clean a cat litter box. And Cryptosporidium comes typically from birds that will contaminate water, and individuals will drink the contaminated water. Again, this is more of a problem for immunocompromised individuals than healthy individuals. The next group we'll look at are the fungi. Fungi are decomposers. They're also very heavily involved in the food industry, but about a third of them are pathogenic. These organisms are also heterotrophs, but they're a special type of heterotroph called a saprophyte or a saprobe. They live particularly on dead or decaying material. Fungal diseases are called mycoses. The fungi have two different types of morphology. There are the molds, which make thread-like hyphae, and a mass of thread-like hyphae is a mycelium, or there are the yeasts, which are single cells. Some fungi are dimorphic. They show both body types dependent on temperature, and your true pathogens are dimorphic. Here you see yeast, and over here is a mold. Here's a mycelium, and here are individual thread-like hyphae. All fungi reproduce asexually, and most reproduce sexually. The yeast produce asexually by budding. They simply pinch off a little piece of the cell, and that cell eventually breaks off. Molds produce spores. Spores are airborne and easily distributed in the environment. Asexual spores are classified on how they are produced. Typically, they pinch off the tips or the sides of hyphae. Sporangiospores, chlamydospores, and cnidospores are examples of asexual spores. Sexual spores result when two mating types combine their DNA. This means that the spores that come off of this union will be different from the spores of either of the original parents. Here we produce either zygospores, ascospores, or basidiospores. Here are some examples of asexual spores. Sporangiospores are produced in a sac, and when the sac ruptures, the spores are released. Chlamydospores are produced when the walls of the hyphae thicken up, and then the hyphae kind of shatter. And then conidiospores are produced off the tips of the hyphae, and you get these chains. Again, as these break free, they become airborne. Sexual reproduction is a little more complicated. You will have to have two mating types, that is, two individual organisms with slightly different DNA. They're usually referred to as the positive strain and the negative strain, and rather than male and female. Now, both of these types live independently, reproduce asexually most of the time, but at some point in time they join. And when you have them join like this, you develop an organism that is dinucleate. It has two nuclei. And it may exist like this for a little while, but typically this dinucleate cell will have the nucleus fuse, and now you will have a diploid organism. Most of the time fungi are haploid. So this diploid cell will begin going through meiosis. Meiosis is going to take a diploid cell and give you 4 haploid cells. These 4 haploid cells will have a different mix of DNA than the original haploid cells they came from. But these haploid spores will go ahead and make a new type of mycelium. They will exist in the haploid stage for several generations, and then they'll go back through a sexual stage again. Classification of fungi is based on how they produce their sexual spores. The division Zygomycota produces zygospores. An example of this division is Rhizopus, which is black bread mold. The division Ascomycota produces ascospores. These spores are held in little sacs and then released. Penicillium, Aspergillus, Claviceps purpurea, and Saccharomyces are examples of this division. Now, Penicillium is the organism that we get penicillin from. Aspergillus is a very common mold. Spores are in the environment all the time, and when people say they're allergic to mold, a lot of times it's Aspergillus. Saccharomyces exists as a yeast, and Saccharomyces is very important to the food industry. This is sometimes called brewer's or baker's yeast because it's involved in making beer and bread. Claviceps purpurea makes an interesting substance called lysergic acid. Lysergic acid leads to hallucinations. It also can cause abortion. Claviceps purpurea may have been responsible for the Salem witch trials. Claviceps purpurea will grow on wheat or rye that has been overwintered in the fields. The year of the Salem witch trials, there was a bad winter and the wheat could not be harvested on time, so it stayed in the field and was harvested later in the year. Probably it had this organism growing on it. As people ingested bread made with the infected wheat, they began having hallucinations. This may explain why people were accused of being witches. What's interesting is that this occurred only in this one winter. After it was over, no one saw witches anymore. And even some of the judges who went to judge the witches wrote in their diaries years later that they don't know why they behaved the way they behaved. It was like something else had taken over. The division Basidiomycota are the mushrooms, puffballs, and rusts. They produce basidia, which are kind of bare. If you take a mushroom and you set it on a piece of paper, and you just let it sit there for several days until it kind of dries out and very carefully lift it, you will see a little imprint of the gills in a fine dust. That fine dust will be the basidia spores. Amanita and agaricus are 2 common mushrooms. Now, agaricus is the nice edible mushroom. Amanita is sometimes called death angel. The 2 look very similar. There's a saying in mushroom hunter world. There are old mushroom hunters and there are bold mushroom hunters, but there are no old, bold mushroom hunters. Because Amanita can look a lot like agaricus, it has been used to poison people. The division Deuteromyces was developed because there were some fungi that had no known sexual reproductive cycle. As we have studied this group more, we have found that most of the organisms that were in this division actually belonged in the division Ascomycota. So this division really has no organisms in it currently. It's pretty much been abandoned. The parasitic helminths and vectors are important to us because they cause disease or they spread disease. The parasitic worms are the helminths, and there are 3 basic groups. The nematodes are the roundworms, things like Ascaris. The cestodes are the tapeworms. Now these are flatworms. And the trematodes are the flukes, which are also flatworms. Roundworms and tapeworms tend to be found in the digestive tract. Flukes can be found in the digestive tract, but they can also be found in other organs like the liver and the lungs. The arthropod vectors are important to us because they transmit disease. There are 2 groups, the arachnids and the insects. Arachnids have 4 pair of legs and 2 body parts. These are things like ticks and mites. The insects have 3 pair of legs and 3 body parts. Fleas, Lice, flies, mosquitoes, and kissing bugs make up this group. And as we study diseases, we will see that these organisms are important in transmitting some of the protozoa.